So, very good morning, all of you. So, this is the welcome to the global lecture series of the Biotech Resource Society. This is the 10th event in this series that we have started in the last 2022 February. So, I request Professor Ashok Pandey for the welcome speech, as Professor Soperi would not be able to join. He's informed due to some personal reason, he could not be able to join this meeting. So I request Professor Ashok Pandey for the welcome address.
Thank you, Vinod, and a very warm welcome to Professor Sunil Kaul. The people in BRSI don't require an introduction about Sunil, who has been associated with the BRSI events and otherwise also is so well known. Not only Sunil Kaul, together with his wife Renu, are so humble and so nice that they have been always appreciated, and with the people wherever they have gone and given talk or attended any program or organized any program. So Sunil is a good friend of course, India, and is doing extremely well for research development. And what is most important, that while we keep talking about in our country that reinventing Ayurveda and trying to do Indian medicine system are trying to identify active components in the medicinal plant which have been traditionally described in India and so on. This has been so well done by Sunil and his group together, and in particular what he's going to talk today is about ashwagandha. I am sure that all those who are working in medicinal chemistry, biological chemistry, and of course infectious microbiology, or whatever we call it, would be of great eye-opening, and information what he's going to share will be of great immense for the all level of researcher. So, Sunil, a very warm welcome to you for this global series, and thanks indeed for readily agreeing to our request to deliver this talk. And at the time, as is normally put 11 hours on 23rd of every month. So you have been very kind to accept it, and we look forward for your presentation. Before that, over to you, Vinod.
Okay, thank you, sir. So Dr. Sunil Kaul is a senior research scientist at the National Institute of Advanced Industrial Science and Technology in Japan, and he has been working in this institute for almost 33 years, and he is also an adjunct professor at Department of Bioengineering, Hanyang University, Seoul, Korea, and he's also a president of Coltech Company Limited, which is a AST venture in Japan. And he's also a chairman, Indian Scientist Association in Japan, and also a research advisor in Rehava Bioscience, USA. And he was also served as an international coordinator for DBT, India-Japan International Laboratory for Advanced Biomedicine. And he was also served as a professor at University of Tsukuba, Japan. And his major research interest is to understand the molecular mechanism of stress, aging, and cancer. He has been merging with the traditional knowledge with the modern technologies like gene silencing and imaging to understand the mechanism of action of Ayurvedic herbs, especially the ashwagandha. With more than 240 research publications in international peer-reviewed journal and several invited talks internationally, he has been the editorial board of several scientific journals. And he has also been honored with several awards, including Fellow of Geriatrics Society of India, Overseas Fellow of Biotech Research Society India, Fellow of Indian Academy of Neuroscience, and Foreign Fellow of National Academy of Science, India. So with this introduction, I invite Professor Sunil Kaul for his talk on overview of ashwagandha, trust and science in Ayurveda. Professor Sunil Kaul.
Thank you, Dr. Vinod, for a kind introduction, and thank you, Ashokji, for your kind words. So let me share my slide. So you have to allow me to share my slide.
Yes, 1 second. Yes. I think you can now share.
So is it clear?
Yes, 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 yes. You
And
can
is
make it in presentation.
presenting
Yeah, yeah,
mode?
fine. This is fine, fine, perfect.
Okay. So first of all, I would like to thank Professor Ashok Pandey for giving me this opportunity to speak in this very important platform, BRSI Global Lecture Series. And before I start my talk, I would like to quickly take you through my academic journey. So I was born and brought up in Kalimpong, a small town in Darjeeling. And from my home, I can see Kanchenjunga, which is 8,586 meters and the world's third tallest peak. So Kalimpong is a hill town. I had my schooling from Kumudini Homes in Kalimpong, and then I moved to Darjeeling to join Darjeeling Government College for my BSc and MSc studies. And from there, I moved to University of Delhi and did my MPhil and PhD, and my research associateship at All India Institute of Medical Sciences, and some work at University of Laval, Quebec. Then came to Japan and joined National Institute of Advanced Industrial Science and Technology, AIST, in '89. And since then, I've been here in this institute. So AIST is one of the major public institute in Japan and only institute for working on industrial science. And we are located in Tsukuba, which is an hour drive north of Tokyo. Now, this is my passion from the start. So when I was in school, I joined Taekwondo as a first batch of Taekwondo in India, and then keep continuing from while moving from Kalimpong to Darjeeling. And then when I came to Japan, I joined Japanese karate and mixed martial art. And this is a photograph in 21. And this is during the pandemic this year and the beginning of this year. And this is just 2 weeks back that I've been continuing all along with my studies or research. Now, so as the title goes, an overview of Ashwagandha, let me introduce Ayurveda. I don't have to introduce, but just for those who are not aware of Ayurveda, Ayu means life and Veda means knowledge. So it is a traditional home medicine system native to Indian subcontinent. Ayurveda prescribes special diet, use herbal medicine, yoga, meditation, philosophy for health care and disease cure. Ayurveda texts begin with the transmission of medical knowledge from gods to sages and then to human physicians. Ayurveda defines human body as three kinds, Vata, Pitta, Pitta, Kapha, and state that balance of these dosas result in health, while the imbalance results in disease. And the Hindu god of Ayurveda is god Dhanvantari, and her birthday falls on October 23rd, which is celebrated in India as Ayurvedic Day. And we also celebrated Ayurveda Day in October last month by organizing an international Ayurvedic webinar. Now, why do we need Ayurveda in the modern world? So with the industrialization and advancement in medical sciences, life expectancy has gradually increased. For example, the average life of Indians are 70, Japan, it is 85. And with this, aging population is also increasing. It is projected that global population is 60 years or over was 0.5 billion in 1990, and it went to 1 billion in 2017, and it is predicted that by 2050, it is going to be 2.1 billion. Similarly, proportion of the world population is 60 years or more were given as 10% in 2000, and it is predicted that it is going to be 21% by 2050. But if you look at 2020, Japan population is 65 and over has already reached 28.4%. That means that the aging demography is becoming a threat. But as we know that aging is not a disease, it is a normal process. And for healthy aging, we don't need pharmaceuticals, but nutraceuticals, Ayurveda, and so that we can decrease the burden of healthcare system. Now, when we do the research on aging or cancer, what we do is we try to develop knowledge reagents, and tools to perform interventions for stress and old age related pathologies. So what we want is add life to years, not years to life. So that means we want to improve the quality of life at the later year of our life. So old age related diseases can be cancer, stress, Alzheimer's disease, Parkinson's disease, sleep disorders, to name a few. So what are cancers? Cancers are just uncontrolled proliferation of cells. It can pop up anywhere in the body, travels from the primary to secondary sites, disrupts normal body function, and reasons could be multifactorial, and mechanism are activation of oncogene, inactivation of tumor suppressor, to name a few. And the treatments could be surgery, radiotherapy, chemotherapy, multiple drug combinations. So now we all know that stress, aging, and cancer have a strong link. As we age, the number of incidents of cancer increases. And by the age 50 or 55, it is more apparent. Similarly, in culture, cells divide for certain population doubling, after which they age and die. But if something goes wrong to one cells, it keeps dividing and become immortal. So in our lab, we want to understand why normal cells age and why cancer cells keep dividing. So we have been looking for factors, molecules that could alter this phenomena. We want something which can extend the life of normal cells, or that one can limit the growth of the cancer cells, that is cancer therapy. So this is just to show how cells look in the tissue culture. This is a normal cell at the population doubling 20. They are very nicely placed and young. They are young cells, but the same cells, when they become population doubling 55, they become flat and disorganized. On the other hand, cancer cells, whether at the population doubling 20 or 120, they look the same and they look young. So in last 33 years, we've been lucky to discover two major proteins through cell culture system. One was a member of a heat shock protein family, HSP70 family, that we named Mortlin in 1993. Since then, we've been working all along. And the other protein we discovered was, we named it as a collaborator of ARF, alternative reading frame, which is a tumor suppressor protein. And both these proteins, Mortlin and CARF, are enriched in cancer cells, and they regulate tumor suppressor protein p53, the guardian of genome. So Mortlin is ready to go for clinical trial. In fact, Oklahoma University, one group has just started clinical trial. But in the meantime, we wanted their inhibitors for cancer therapy. So 15 years back, we moved to Ayurvedic approach and screened a battery of library of chemicals and phytochemicals, including lots of vidinolides found in ashwagandha also. And what we found that Vidania somnifera came in our top list. So Vidania somnifera, which is commonly known as ashwagandha, also known as Queen of Ayurveda, we started focusing on. And why ashwagandha? Because ashwagandha has been in use in Indian subcontinent for different ailments for hundreds of years, for increasing memory, treating insomnia, stress, hypertension, diabetes, arthritis, and so on. But the problem is mechanism was not clear how it works. So we wanted to know whether ashwagandha actives can be used for intervention of stress and cancer. So what we did, we took ashwagandha leaves. In Ayurveda, ashwagandha roots have been in use. So we wanted to know what is there in the leaves and other parts of the plant. And also when we take leaves, we don't have to sacrifice the plant, and also we can have an ample supply for our experiment or for the use. So what we did, we made an extraction, alcoholic extraction, water extraction, and characterize all the components present in there and put it in our in vitro cell culture system. So here, we always compare the disease cell and the normal cells. For example, if we are looking for something that is to be cancer therapy or that inhibits cancer, we also use normal cells to see what it does to the normal cells. So once we get the targets, we move on to the rodents, and we use mouse or rats for this purpose. So what we found, that we made a ashwagandha leaf extract, we called it eye extract. Then when we treated the normal and cancer cells, what we find that eye extract or the ashwagandha leaf extract kills cancer cells, but nothing happens to normal cells. This is to show you here that these are normal cells, these are cancer cells. When treated with ashwagandha extract, normal cells keep proliferating, but cancer cell dies. So when we look at the in vivo studies, so when we inject fibrosarcoma, very aggressive cancer, to this nude mouse, it gives a solid tumor by day 12. But if we inject eye extract at the same time, then there is a strong inhibition of tumor. This was very interesting for our studies. So this is just to show that how we study nude mouse or the tumor formation. We inject the tumor cells or cancer cells either by tail vein injection or subcutaneous tumor or intracranial injection to nude mouse, and we make the tumors. And then we inject our test material either before or after to see the prevention or therapeutical efficacy. So once we knew that ashwagandha leaf extract, that is eye extract, kills cancer cells, we wanted to understand the mechanism. So we took three kinds of approach. One is called loss of function screening. So what we do here, we take a 96 well plate and then culture the cells and then transfect them with siRNA library in the presence of eye extract or particular vithanolite or components. So whatever the viable cells are there, we find the target. Similarly, we use a randomized ribozyme library screening. This is a blind system. Here also we find the targets. So whatever targets we get, we take it to the third approach. This is a bioinformatics approach. So by this method, we can know that what are the molecules, whether they are aligned or they are in some networking. And what we found that ashwagandha leaf extract or eye extract kills cancer cells by activating p53, p21 pathway, which is a tumor suppressor pathway, or by activating oxidative stress. So that means ashwagandha alcoholic extract, major component vithanone and vithafurin A, kills cancer cells by activating tumor suppressor protein p53. So once p53 came to our screening, our studies, then we were excited because p53 is also known as guardian of genome. It controls cell division, and in more than 70% of cancers, p53 is inactivated or it is mutated. So we wanted to validate whether it is true that p53 is involved. So we took a biochemical approach. Here, what we do, we treat the cells, normal cells and cancer cells, and then put it through electrophoresis, and with the Western blotting specific to the antibody, we try to see the expression. So here we are looking at p21, which is downstream of p53. We see here that in response to eye extract, only in cancer cells p21 is upregulated here, but normal, there is nothing here. So which means cancer cell growth arrest, or there's a tumor suppressor. Now, once we knew that ashwagandha extract is killing cancer cells, we went one step further to look at the cancer cells which are very difficult to treat. So here I would like to introduce telomeres. Telomeres are the ends of chromosomes and that shorten with each division. But in cancer, telomere length is maintained by activation of an enzyme called telomerase. So if we have an inhibitor of telomerase, we can stop the cancer growth. But this is there in 95% of cancer. But in 5% of cancer, the telomere length is long, but there is no telomerase. And the length of telomere is maintained by another mechanism called ALT mechanism, alternative lengthening of telomeres. And these difficult cancers are difficult to, they are resistant to telomere inhibitors and difficult to treat. So we wanted to know that these vithanolites found in ashwagandha has any effect on these ALT cancers also. So we made isogenic human cancer cells and identified telomerase plus cells and ALT cells and treated them with vithanolites, for example, vithafurin A. So what we found that Vidaferin A kills telomerase positive cells by growth arrest, by activating p53, as I said before, but it also kills all cells by apoptosis. So that means vidaferin A inhibits alt mechanism of telomere maintenance and induces apoptosis in all cancer cells also. So this is a story when I'm talking about alcoholic extract that we use, I extract, but we were thinking if we want to use ashwagandha in water just like that, so what happens whether these kind of bioactives are still there and whether they also take this pathway, what happens? So we made a water extract of ashwagandha and we went through the battery of experiment that we did with the alcoholic extract. And what we found, interestingly, that in water extract, the active component is not vidanone, not vidaferin A, but it is a triethylene glycol, small molecules. So we checked again with triethylene glycol or water extract if it is killing cancer cells specifically. Yes, it kills. So what that means is we made an alcoholic extract. The active component in alcoholic extract are vidanone and vidaferin A. These are vidanolides. And it kills cancer cells by activation of p53 pathway or by oxidative stress. But water extract has a triethylene glycol, not vidanone, and it activates RB pathway, retinoblastoma pathway, which is an alternative tumor suppressor pathway. So if we have an extract that has vidanone, vidaferin A, triethylene glycol, it could nicely manage the tumor. But if we are going through one pathway, if we are making a one type of extract, we are only taking one pathway and we are not touching the other pathway. That becomes incomplete. So this is the reason why in Ayurveda, suggests that the whole herb or grass extract is important, not purified single compound. Now, as I mentioned before, that ashwagandha kills cancer cells, but nothing happens to normal cells. But we were curious to know what happens to normal cells then. So we took 3 types of human normal cells, WI38, TIG1, MRC5 cells, and treated them with I extract, that is alcoholic extract. And we see here that the normal cells are growing very nicely, but cancer cells, this is a bone cancer, U2S cells, are killed as mentioned before. Vidanone also shows that normal cells, nothing happens to normal cells, they are dividing very nicely. Vidaferin A, on the other hand, kills cancer cells, but it is a little bit toxic to normal cells. But the interesting point is, if we treat vidaferin A, which is a bit toxic to normal, in the presence of vidanone, then this toxicity is lifted. That means we have a vidanolide that kills a little bit toxic to normal, at the same time that protects the normal cells. And in another approach, what we did, we took a normal human fibroblast and treated with MAA, which is a toxic metabolite of ester phthalates found in all kinds of plastics that we use commercially. So when we treat the normal cells with MAA, we find a premature aging of cells, DNA damage, oxidative stress, mitochondrial damage. But if the treatment is in the presence of this alcoholic extract of ashwagandha, then viability is better. There is no premature aging, DNA damage is less, no oxidative stress, and there's no mitochondrial damage. Likewise, if we treat the normal human fibroblast with another type of oxidative stress, that is treating with hydrogen peroxide, number of cells you find here less than the control here, but in the presence of extract, the number of cells are better. So that means it is protecting these normal cells against oxidative stress. Similarly, UV damages the cells. Here we see the number of colonies less compared to control, but in the presence of extract, number of colonies are more. So that means ashwagandha extract is protecting the cells against oxidative stress, against UV damage. And now we wanted to see what happens to the cell population. As in my earlier slide, I said that the cells divide for certain population doubling. After that, they age and die. So we wanted to see the effect of these vidanolides or the extract. So we cultured them to look at the population doubling in these normal cells, which is a 4 to 5 month study. And what we find, this blue line, the cells are going up to this population doubling, and then they stop dividing. But if the cells are there in the presence of vidanone, they go another 5 to 7 population doubling. In terms of human cells, it is quite significant. And this means that these cells are getting lifespan extended. That means the aging is delayed here. So vidanone and extract are good for normal cells. Let's see what they do to the brain-derived cells. That means anti-stress and neurodegenerative potential, whether they have. So old age-related pathologies, as I mentioned before, they are Alzheimer's disease, Parkinson's disease. And if you are looking at Alzheimer's disease or Alzheimer's brain, we think about loss of neurons, loss of memory, accumulation of oxidative damage and amyloid plaques. And all these can be studied in laboratory by treating them with chemical models, by treating them with hydrogen peroxide, glutamate, scopolamine, or making the rat or mouse MTTP mouse. So we treated neuroblastoma cells or glioblastoma cells with oxidative stress, excitoneurotoxicity, amnesia stress by giving them hydrogen peroxide, glutamate, scopolamine, and all these challenges put the cells into death, apoptosis, or loss of differentiation markers. But if we treat them in the presence of extract, there is no cell death, no apoptosis, and loss of differentiation marker is not there. Rather, cells start differentiating. So in another study in collaboration, we found that this is a mice model of Parkinson's disease. Here, what our collaborators are looking at different parameters like rearing, grooming, time taken to cross the beam, and foot slipping errors by this Parkinson model of mouse before and after they were fed with ashwagandha extract. And what they found that all these parameters have improved after this mouse was fed with ashwagandha, like grooming, time taken to cross the beam is corrected, foot slipping error is also corrected. And very interestingly, if you look at the footprint, this is a footprint of the control mouse, and this is a footprint of the Parkinson's disease model mouse. We can see wobbly movement here, but once they are fed with ashwagandha extract, the movement is much corrected. We can see this is better than here. And another study, we looked at Drosophila, which is also a Parkinson model Drosophila. And these flies were, these are the Parkinson model flies, and we look at the climbing activity in a test tube. We find that they go for certain distance and they cannot move further, or some are lost at the bottom here. But when they are fed with the water extract of ashwagandha, these flies are going much faster, and they cross the ring and goes up. And when they are fed with the water extract 10%, they are much, much faster. And they are not only crossing the first ring, also they are going to the second ring, and they are now here at the top of the test tube, which means that ashwagandha extract has a potential to help Parkinson's disease. So that means ashwagandha says that may improve brain function, including memory. We say from our experiment, yes, it does. Now, we were trying to characterize all the components present in ashwagandha. So some examples like vidanone, which has a moderate anti-cancer activity and it kills cancer cells selectively. On the other hand, vidafrin A has a strong anti-cancer activity, but it is a bit toxic to normal cells. While we were screening other vidanolides, we came across 3-beta-methoxy-vidafrin A, which is very similar to vidafrin A, but there is a change in methyl group here. So we were expecting that this could be a better use for cancer treatment, but to our surprise, that vidafrin A, when we treat the cancer cells and look at P53, oxidative response, DDR, we find them going up and migratory properties going down, and the result is cancer growth, cancer cell growth arrest. But 3-beta-methoxy-vidafrin A, when we look at this parameter, there was no effect. So 3-beta-methoxy-vidafrin A with just one little change has no effect on any of these parameters. So we try to see then what happens to normal cells then. So we again treated normal cells in culture with vidafrin A, and 3-beta-methoxyvidafurin A. And we find that vidafurin A at a higher dose kills normal human cells, but 3-beta-methoxyvidafurin A does nothing to normal cells. And we went on to see the response of this vidafurin A and 3-beta-methoxyvidafurin A after giving the cells different kind of stresses. For example, oxidative stress, treating them with hydrogen peroxide, UV, or doxorubicin, cisplatin, etoposide. And after this treatment, we put them in vidafurin A or methoxyvidafurin A. We see that in the presence of vidafurin A, these cells after the treatment of doxorubicin are dying further. But 3-beta-methoxyvidafurin A not only protecting, but they are helping in recovery. Similar results are found in response to other stresses also. So that means vidanone has a similar structure, has an anti-cancer potential, but vidafurin A has a side effect on normal cells, but at the same time 3-beta-methoxyvidafurin has a side benefit. That means in nature there are components which are bad for cells and there are components which are good for cells. Now the question is, when we are using ashwagandha, we have to be very careful that what kind of ingredients, bioactives are there and what purpose. If we are find ashwagandha with only vidafurin A, this must be maybe killing the cells, cancer cells, but it also affecting the normal cells. Similarly, 3-beta-methoxyvidafurin may not be helpful for treating the cancers. Now, when we are talking about stress, we cannot go without talking about SARS-CoV-2 pandemic, as we are still going through. It started in 2019 and 2020 was the peak. And by now, there are 697 million cases of COVID positive and 6.7 million deaths. So our interest was to look at, again, screening the phytochemicals, including vidanolides, and see if any of these can help in either prevention of COVID or the infection. So if you look at the steps in virus life cycle, so when virus comes to the normal human cells, it attaches, penetrates, and then it takes the machinery of the host cells. Biosynthesis goes on, maturation is there, and then when comes out lysis, and then it attacks other neighboring cells. So our interest was to look at different chemicals, including vidanolides, and our target was to target these receptors. One is a ACE2 receptor, one is TIMPRESS2 receptor, and also AMPRO3. So for this, we did extensive computational analysis in collaboration with Dr. Durai Sundar from IIT Delhi. What he did was he put these vidanolides all in his binding assays for computational. And what he found that many of the vidanolides show good binding to ACE2 protein. And then we look at this expression in culture and find that, yes, indeed, these vidanolides suppresses ACE2. So he also looked at these vidanolides against TMPRSS2 protein, and he found that this also has good binding potentials. And we also look at its potential for its binding to AMPRO, molecular docking analysis. And here also we find that some of the vidanolide bind to AMPRO very nicely. And we published this paper in 2020 at the peak of the pandemic. And so what that means is ashwagandha has inhibits virus cell surface receptor binding, that is ACE2 and TIMPRESS2. And by in vitro wet lab studies, we saw that there is a downregulation of cell surface receptors at mRNA and protein level. And we also see binding to the catalytic site of viral protein AMPRO. So now, after this is the computational and in vitro studies. So we really wanted to see whether it is active against the real virus. So we did a collaboration with Professor Sudhanshu Bharati from Regional Center for Biotechnology. What they did, they used VeroC E6 cells and infected with the virus. This is a control here. we can see viral replication. And then in the parallelly, he put this test molecules here. And in summary, what they found that ashwagandha leaf extract has an inhibition rate of 90%. Here they are using positive control as remdesivir, which has 99.76% capacity for prevention, inhibition. And other vithanolides also show less or more activity. So that means the take-home message is ashwagandha has multiple activities, including anti-cancer, anti-stress, anti-aging. While some bioactives show side effects, others have side benefits. And the Ayurvedic approach says use the whole herb or grass extracts, not purified single compound. And we saw from our experiments, indeed, the whole herbs or grass or extract is important for to take it as a holistic approach. And in addition, ashwagandha has potential to be a natural resource for managing COVID. Now, the point is, we have to we need a defined standardized extracts and products with defined vithanolides for particular treatment. For example, if we are looking at treating cancer, we have to have a ashwagandha with high amount of vitamin A and some other vithanolides in combination. And for that purpose, and all the plants has to be standardized from their cultivation to the making the extracts. So we, in collaboration with Guru Nanak Dev University, with Professor Pati, been studying in vitro cultivation of ashwagandha in greenhouse plant. Also, we are making, then breeding vermicompost in Japan. We are growing ashwagandha organically in sand with as a sand ponics, water ponics. So this is one example. We've been successful in establishing ashwagandha in hydroponic in collaboration with Osaka Prefecture University and Daidan. So our aim is to bring ashwagandha with defined vithanolide or bioactives for defined disease and the defined disease. So every ashwagandha, two ashwagandha plants found in two regions are totally different. And also we saw here that not only the root, but other parts of the plant are more or equally important. So with this, I would, so this is the book we published a few years back on science of ashwagandha, preventive and therapeutic potential, in addition to the publications. And I would like to thank all the colleagues, students, and collaborators who have contributed to this project. I'd like to thank Regional Center for Biotechnology, Guru Nanak Dev University, IIT Guwahati, IIT Delhi, Sikkim University, Manipal University, University of Tsukuba, and special thanks for this project to Dr. Sundar from IIT Delhi, Gurucharan from Guru Nanak Dev University, Professor Pati from GNDU, Dr. Redl for telomerase and cancer studies, Professor Chiok Yoon for in vivo studies, and Dr. Terao for cyclochem. And with this, I would like to stop here and take any questions that you may have. Thank you so much for your attention. So, shall I stop sharing now?
Yes.
Yes.
Hello.
Yes.
Thanks, Sunil. We can take some question answer if you have time.
Yes, sure. Yes, please.
So, audience, if you have any question, please put on chat box and we will take them up.
Well, on set of this, I thank Professor Sunil Kaulji for beautiful presentations. And we have been knowing that he's been working on the ashwagandha for so many years, and even he has given me that ashwagandha, which I'm taking daily. Ashokji, I think this work is very important. Then what they have found is the most important thing is the small biomolecules, what we call the untargeted metabolites. So these are the compounds which are very important, as got a very, very potential, all the immunomodulators and all. And they are purified, they are characterized and all. Now the thing is that we are now thinking to supplement it as a food. The concept of the Ayurveda as a Ayurveda ahar, that is what is the food as medicine. Food as a medicine we don't have to take. Nowadays, in the olden days, we take the food as a medicine. Every disease, every traditional food, every ethnic food have got the therapeutic uses. But now it is a separate. Food is different for the taste, and we take the extra medicine. Now I think the concept has to come with the olden days, the ancient times, the concept of the Ayurveda ahar. So in that way, I think in future we are thinking in that way. Probably maybe by next year we come with the proposals to make some traditional fermented food or some non-fermented food mixes with this ashwagandha. This is what this is my suggestion on behalf of Professor Kaul. Am I right, Professor Kaul?
Yes, thank you, Professor Tamang.
Sunil, there is a question that how about the stability of the extracted in water and alcohol to temperature and pH or any other parameters tested?
Yes, we do the extraction at high temperature, but it works in high temperature as well.
And there is another question.
And
How among,
yes, yes.
sorry,
Please.
you can see it?
Yes.
How
yes.
among all the metabolites in extract, did you understand that ethanol are of prime importance?
Yes. So what we found that if we are looking at one particular disease, so one particular component or the combination of other component are very important. If that particular bioactive is not present, it doesn't work. And likewise, we had a collaboration with Tsukuba University this Sleep Institute, where they found that triethylene glycol, which is important for sleep, not other vithanolides. So this was a very surprise finding. So if the triethylene glycol is present in ashwagandha, it helps in sleeping. So Vidania somnifera itself means sleep inducing, or it helps in sleep. But if triethylene glycol is not there, it may not work. So we have to be sure that the ashwagandha that we take for sleep must have this component. So we have to define this when we are using it.
So, hello, Professor. Can I ask, sir?
Sure.
Yes, please.
Hello, sir. Very nice presentation. I have small doubts, sir. Nowadays, some publications are arriving or emerging in the translation research journals regarding the nano ashwagandha. I think you know it, because they have the time-lapse releasing profiling of the ashwagandha crude with the nano profiling during the bioavailability. What is your opinion, sir?
Yes, so we have done that work for cancer treatment. We have used folic acid receptor, looking at folic acid nano capsules, we made it. And then the cancer that has a folic acid receptor, only this nanoparticles goes into those cancer and it targets those cells very nicely. So this is a way of delivery, and I think we have been successful in that. So it is. But then again, the question is not the one particular compound, but whole extract.
Correct. No, what I'm saying is just a hypothetical view. In the bioabsorption rate during the digestive modes,
yes,
when we are having the pH, different pH coefficients in the GI, during that one, does this nanoparticle or nano-formulated ashwagandha have some de-efficacy syndrome or a phenomena, sir? Any
no, we have not done physiological study or in vivo human things, but as far as we have done in vitro with nanocapsulation, it works.
Correct.
For others, we haven't done it. So we have no idea what is the consequence or other effects. But idea is to increase the bioavailability when we are encapsulating.
One small last question. Sorry for the whole thing. Is there any particular receptor for this Withania? Not an individual, with the whole Withanic compounds in the epithelial cells or lining cells?
We haven't looked at the receptors, but there is a differential internalizing of these molecules in cells. For example, Professor Sundar, what they have done by computational, by simulations, that Vidanone goes into the cancer cells very nicely, but it does not pass.
Right, correct, that's what.
The cancer cells. So that way we can differentiate. Also, we did some bioimaging to see that Vidanone or Vidafrin A, which one goes earlier inside the cells using quantum dots. And likewise, we find that there's a differential permeability in the membrane or internalizing of the molecules. So we raise the antibody against these molecules also, Vidanone and Vidafrin A. Not very easy, but we have that, and we find that there is a difference. That's how it becomes specific to cancer, but not to normals. It does not kill normal cells. So there must be some receptors or some permeable membrane changes are there in the cancer cells that allows these molecules to go in very easily.
Thank you, sir.
Sunil, there are a couple of questions, and there is a comment also.
Yes.
One question is cultivating ashwagandha in hydroponic condition. Can you produce similar amount of bioactives as compared to cultivating in field conditions?
So idea of doing hydroponic cultivation is to make it in a very controlled environment, so we don't have a contamination from any other things like maybe soil, microbes, or other things. It's a very defined way of cultivating, so that the ashwagandha that we get it will have all the same particular compound in all ashwagandha very uniformly. Otherwise, what we see that in the field, that ashwagandha from one place and another place, they are so different. So we are trying to clone them, but it is quite difficult. So that's why hydroponic or sandponic, these are the methods to have a standardized, or make a ashwagandha cultivation standardized.
There's another question that is quite interesting. I was curious if we, general public, can take ashwagandha in our daily diet. If so, with what standardization and in which form it can be taken?
Our ashwagandha is still not in market, or we have not commercialized. We're still working for last 15 years. It will take some more times. And although we are we have now completing the first clinical trial in US for lung cancer, only that part is already there. First clinical trial is finishing this month. But besides that, our ashwagandha that we have a defined one is still not in market. So what is available in market, we have to be careful that which ashwagandha with what is there in there. So we are, last month we signed a MOU, our institute has signed a MOU with Ayush Ministry to do all this analysis of all the phytochemicals or Ayurvedic plants, medicinal plant, to do molecular mechanism, how we are doing, just following ashwagandha as one model.
There is general comment making hydroalcoholic extract of ashwagandha leaves could be a good approach rather than making alcoholic and water extract alone.
Yes, I agree that. But again, we have to be very careful that, as I mentioned in our, what we found, that alcoholic extract is killing cancer cells by activating one particular pathway, not, but the water extract is taking another pathway. So we have to be careful whether all these pathways are included in that extract. So we have to test that, but we haven't done any other except alcoholic and water extracts.
There's another question, Sunil, that can the product after purifying be used as a herbal-based medicine?
We prefer to make a botanical drug rather than just taking an individual component and mix them. We want to take a ashwagandha plant or extract as such, but the compound which is bioactive, what we want should be in a higher proportion, in a particular, at least particular proportion. Then it is, we are satisfied that it is working for that particular disease. But if that balance is not there, then it doesn't work. That's what we have found.
I think what you're saying, Sunil, is largely true for success or failure of Ayurveda, what we have seen in this country. Unless there is a counted quantity, minimum quantity of bioactive component reaching into body,
Yes.
perhaps the impact may not be there. And that is why perhaps there is a trust or distrust on Ayurveda in some part of the country, and many other part of country, there is more distrust. So do you think that this could be one of the possible reason that people taking it in a raw form where bioactive molecule may not, bioactive components may not be even stable because storage condition and all those things will impact the bioactive components in any even storage of period of storage. For example, something is stored for 2 years, perhaps may lose its volatile component, whatever, you know. So do you think that this is one of the important aspect for Ayurvedic research in general or ashwagandha in particular? One should keep into mind.
Yes. So when we started, we collected ashwagandha about 35 places, and all of them are different in terms of their molecules characterized when we did the characterization. In some cases, only vidafrin A is very high, and some cases vidanone or like that was found. So if we are taking vidafrin A ashwagandha, maybe this is a little bit toxic to consumption. And similarly, if vidanone A is very high, vidafrin A is not there, then it is no use for the cancer. So that's how there was so much difference because of the secondary metabolites, soil condition, water condition, places. So we have to standardize them and put them together, and then only use it. So that way we will.
Can I comment, Ashok?
Yes, please.
Sure, yes.
Welcome, Professor Singh.
Sorry, I have missed this because I was into something else. It is a very, very interesting topic for me. I think it's very exciting to talk about these things. But do you have any idea the mechanism of action? You have multiple compounds. You think that they bound to something which is related to cancer, or even they will bind here and there. They are not very specific to a particular target. So how do you see the mechanism of action using such materials? Is my question clear?
Yes. So that was all about the talk today.
Oh, you talked about this already.
Yes,
I missed that. Sorry.
So what
Okay.
Briefly, I will say.
No, don't go and repeat this. There's no point.
So
now,
we characterize
the point just to take from what Ashok was talking.
Yes. So what we did was
Sunil, listen.
Yes.
I don't want you to repeat that.
Okay.
The point is that
yes.
See, you cannot expect plants to have specific molecules for your treatment for human beings.
Yes,
but they may provide some kind of little lead kind of molecule, and from that eventually you have to go back to chemistry to make them better and more potent and more useful. Yes. Are you thinking on those lines?
We are not interested in going for a reductionist approach, again purifying one compound and going for particular disease, but we are looking at the holistic way how the plant with a different combination could be helpful in particular disease. So as my yes.
So you essentially want to stay alive with a specialist, not as a scientist addressing the real problem of making more potent substances.
So there are compounds like taxol, cisplatin, all those things already plant derived. So we are not going in a reductionist approach. In our studies, we are trying to grow ashwagandha for example, ashwagandha with particular bioactives that could be help for particular problem, but not others. And we've been so far, and that becomes safer also. And so far we are going good actually. Our data and our work shows that we can do that.
The question is that you don't want to do things the lab, you want ashwagandha to do this for you.
Yes.
So you want to try new varieties of ashwagandha and something like that, or what?
No, we have established particular kind of ashwagandha with particular bioactives, and we are not generating new kind of ashwagandha, but from the ashwagandha that we have by inbreeding, we have cloned ashwagandha that has different activities.
Good. I think this is one approach, very interesting.
So that means we need the plant with particular methanolite signature that works for particular problem.
Okay, good. Good, very good. Okay. So I think, Shok, I can publicly congratulate you today, and we are face to face.
Yeah.
So
we are not.
Yeah, but what I feel that this could be a
Thank
safer
you.
cancer therapy, because if you are using a particular chemotherapy.
Sorry, Sunil, I was saying something else.
Sorry.
Okay. Good.
Okay.
Sunil, please go ahead.
Very
Sunil,
nice.
please go ahead.
Yes, please go ahead. Yes.
Sunil, let us continue with you, and then we'll come back to processing.
Sure. Yes, please.
You said that you're thinking of therapy or something?
Yes. So our first set is already in a clinical trial, which is completing first phase with FDA approval in USA, in Ohio. And we are now waiting for the investors for going to the second phase, which is very expensive, but we are waiting. So this is for small cell lung cancer therapy. So with that, we may go for other cancers and cancers also. So this is a one part. And other part, now we'll start working with Ayurveda Institute very soon on ashwagandha and other herbs that they are interested in. So idea is to follow the study what we have done with ashwagandha to go for other herbs also.
Anybody, any of the audience participant has any question to Sunil? Any query? Otherwise, move to the second part of Q&A. All right, Sunil. So I have a question to you that is.
I was thinking that there is something wrong with the starting, not what.
So this normally to all the global series speaker, I take the privilege of asking this question. So I'm going to ask to you that starting from your days of school, when you're in high school, did you dream to become such a important scientist, to go into doing research of on a topic which you have so great relevance? How did the events happen? We completely change your life, and from all that, what is your suggestion, advice that which may inspire, or which the people who are attending it you like to share, so that may help them, or that may inspire them, or that may be a kind of guiding force for them to adopt, so that they also become a great scientist, great researcher, great human being like you?
I think I come from a very humble background, from a small town in Kalimpong, as I introduced from earlier. So hard work is very important for us there, and I think for everyone, and consistency that I have followed. But yes, I'm very happy with what I'm doing in research that is directly helpful to the society. And when in my school days, when at home when we were talk about Ayurveda, or somebody used to talk, I never believed. I thought the plants like Ayurveda or plants are nothing, actually it is useless. So we go for even if for a little bit of headache or something, we used to go for anacin or paracetamol rather to going for taking care by this kind of herbal treatment. So, but when I started working, when I started work in stress, aging, and cancer in molecular biology with that background, when I started working on ashwagandha, it completely changed my perception. And now I really believe that there is something which is very, very important, economic, and it comes from our ancient teaching, which is not just like that, it is true. But only thing is we have to be careful how we can make it evidence-based so that people will believe it and then use it for our good quality of life. So my suggestion, this thing is only is consistency and hard work to go on with what we are doing, and then have passion with whatever we do.
Thank you, Sunil. Thank you so much. So, Vinod?
Yes.
Vinod?
Yeah.
So, I think we can wind up. Before that, we can invite Professor TP Singh to say a few words, and then we can formally close it. Professor TP Singh?
Where is Professor Sudhir Sapori?
Professor Sapori has been unable to join today due to some personal inconvenience.
Okay.
So he informed in the morning that I will most likely not be able to join.
Okay. So very nice. I am also sorry that I missed, because yesterday I wasn't here. So I got involved in something which could not be over before 11:30, 11:45. So it's my loss. I think there can be so much discussion, so much excitement on this natural, particularly plant products. And perhaps this will strengthen existing Ayurvedic practices. So I think what Sunil has presented in, I assume what he has presented will make progress in that direction. So it's very good in that sense. And I think sometimes it would be nice if you listen to him face to face.
Yeah, we'll ensure that Sunil is there in the next BRSI meeting.
Sure.
Next means in December or next time?
Next time, not this one. Yeah.
Not this one, next
this
time.
one.
Excellent.
Yeah, so that will give opportunity to all of us to talk to him on a lot of these things. But this subject is such that there are lots of ideas on this. And he uses a term reductionist approach, but I think a more appropriate term would be that you have to make more specific molecules to treat deadly diseases, otherwise they don't get cured easily. So that is the thing. So nevertheless, I think it's all the best to you and congratulations. And I'm very delighted. And I must share, I will share afterwards. I think I should not share just now. I discussed with something about Professor Ashok Pandey. So should I tell others or not?
You can.
You are stopping me.
No, nobody is stopping you. Nobody is there who can stop you.
No. It's good. We will talk it later. Okay, Vinod, take over.
Yeah.
Do things.
Okay. So thank you, Professor Singh. So at the outset, I would like to thank Professor Sunil Kaul for this very wonderful and very, very informative talk. And it was really very, very interesting and really an important area of research, which would be benefiting and to improve our human life and disease condition. So thank you, Professor Sunil, for taking time and joining in this meeting. Thank you very much.
Thank you.
I also thank Professor TP Singh and Professor Ashok Pandey and all other members who joined in this meeting, in this global lecture series. So the next lecture in this series would be in the month of December by Professor G.D. Yadav. So this notification will be released soon about the topic of his talk. So with this, I think we can wind up this series, and thank you once again all.
Thank you so much.
All the best.
Thank you.